In vitro gene cloning, the polymerase chain reaction. The polymerase chain reaction is a method of copying fragments of DNA. The process is automated, making it both rapid and efficient. First of all, you need a DNA fragment to be copied. DNA polymerase, which is an enzyme capable of joining together thousands of nucleotides in a matter of minutes. One such enzyme, called Taq polymerase, is obtained from bacteria in hot springs and is therefore tolerant to heat. It's thermostable and does not denature during the high temperatures as part of the process. Primers, short sequences of nucleotides that have a set of bases complementary to those at one end of each of the two DNA fragments. Nucleotides, which contain each of the 4 bases found in DNA. Thermocycler, a computer-controlled machine that varies temperatures precisely over a period of time. The polymerase chain reaction occurs in 3 stages. Separation of the DNA strand. The DNA fragments, primers and DNA polymerase are placed in a vessel in the thermocycler. The temperature is increased to 95 degrees, causing the 2 strands of the DNA fragment to be separate due to the breaking of hydrogen bonds between the 2 DNA strands. Addition, or annealing of primers. The mixture is then cooled to 55 degrees centigrade, causing the primers to join or anneal, forming hydrogen bonds to their complementary bases at the DNA fragment. The primers provide the start sequence for the DNA polymerase to bind to begin DNA copying, because the DNA polymerase can only attach to nucleotides to the end of an existing chain. Primers also prevent the 2 separate strands from simply rejoining. Synthesis of DNA. The temperature then increases to 72 degrees centigrade. This is the optimum temperature for the DNA Taq polymerase to add complementary nucleotides along each of the separated DNA strands. It begins at the primer on both strands and adds the nucleotides in sequence until it reaches the end of the chain. Because both separated strands are copied simultaneously, there are now 2 copies of the original fragment. Once the 2 copies are completed, the process is repeated by subjecting them to the temperature cycle again, resulting in 4 strands. The whole temperature cycle takes 2 minutes. Over 1 million copies of DNA can be made in the 25 temperature cycles, and 100 billion copies can be made in just a few hours. The PCR reaction has revolutionized many aspects of science and medicine. Even the tiniest sample of DNA from a single hair or speck of blood can now be multiplied to allow forensic examination and accurate cross-matching. Advantages. It's extremely rapid. Within a matter of hours, 100 billion copies of a gene can be made. There is no loss of valuable time before forensic analysis and matching can take place. A complicating factor is that PCR will also increase massively any other contaminating DNA found at the scene. In vivo cloning would take many days or weeks to produce the same quantity of DNA. It does not require living cells. All that is required is a base sequence of DNA that needs amplification. No complex culturing techniques requiring time and effort are needed. The advantages of in vivo cloning are: it is particularly useful where we wish to introduce a gene into another organism, as it involves the use of vectors. Once we have introduced the gene into a plasmid, the plasmid can be used to deliver the gene to other organisms. It involves almost no risk of contamination. This is because a gene that has been cut by the same restriction endonuclease can match the sticky ends of the open-ended plasmid. Contaminant DNA will therefore not be taken up by the plasmid. In vitro cloning requires a very pure sample because any contaminant will be copied. It is very accurate. In vivo copying, the DNA copied has few, if any, errors. At one time, about 20% of the DNA cloned in vitro by PCR was copied inaccurately, but modern techniques have improved the accuracy of the process considerably. However, any errors in the copying of DNA or any contaminants in the sample will also be copied in subsequent cycles. This problem hardly ever arises with in vivo cloning because although mutations arise, they're very rare. In vivo, you cut at specific genes. It's therefore a very precise procedure, as the culturing of the transformed bacteria produces many copies of the specific gene and not just copies of the whole DNA sample, like in PCR. In vivo, it produces transformed bacteria that can be used to produce large quantities of gene product. The transformed bacteria can produce proteins for commercial or medical use, for example, insulin.